Okay, I got this question from one of our student, and this question is: a scientist is studying a population of lizards with three different color phenotypes. The color phenotype is controlled by a single gene with two alleles: an incompletely dominant allele A and a recessive allele A. The scientists collected the data on the color of 100 lizards. That the data are shown in the table. Now, first of all, you must know that it is a case of codominance or incomplete dominance, in which the AA is going to represent a different phenotype, this is going to represent a different phenotype, and this is going to represent a different phenotype. Red, purple, and blue, they are showing you there. And when they did the experiment, they found out that red color is shown by 75 individuals, the purple is shown by 15, and the blue is shown by 10. So this is the question you can see over here. They are saying to test the hypothesis that the population is at Hardey Weinberg equilibrium, the scientists perform a chi-square test. Okay, now when you need to find out a chi-square test, first of all, you need to find out the observed ratio, expected ratio, because obviously these are the groups. If you look closer to the chi-square test, these are the groups, and these are the observed values, and then you are supposed to know the expected values. Then there would be the table for O minus E, then there would be O minus E whole square, then there would be O minus E whole square over E. So let me tell you, now how are you going to find out the expected value? For this purpose you need to understand the Hardey Weinberg equilibrium. You know that in Hardey Weinberg equilibrium, P is equal to the dominant allele, which in this case is A, and Q represents the recessive allele, which in this case is small a. Now if I ask you to find out the frequency of P, dominant allele, and find out the frequency of recessive allele, you first have to realize this thing. That when we do the Hardey Weinberg equilibrium, we say that P plus Q equals to 1. So to find out the probabilities of P and Q, you must first realize that your value must be between 0 to 1. So let's say, if I say that 75 lizards are showing the red color, means capital A. So what I would say, that the red color is shown by 75 over 100 equals to 0.75. Okay, you have to remember that you have to keep your value between 0 to 1. So to put them, or to just adjust them between 0 to 1, you have to divide it with 100. And let's say how many are showing the blue color? They are 10. So 10 divided by 100, that would be 0.1. Now the problem is that when you represent P is equal to A, then how do you represent AA? You would represent AA by P square, not by simple P. The single P represent one capital A. What does it mean? If single P represents one capital A, then two P's would be representing two capital A's, which would mean P square. For your satisfaction, let me write it out. Homozygous dominant, capital A means capital P. One P represents this P and the other P represents this P, so it would be P square. And then when you talk about the homozygous recessive, small a, they are not going to be represented by Q, because you said that Q represents the recessive allele. So if there are two recessive alleles, then you would write QQ, that would be Q square. Okay, now I need you to find out the frequency for the dominant allele and frequency for the recessive allele. The problem is that in case of frequencies, you need to first consider that capital A shown by red, the reading for capital A is 0.75. So what does it mean? That if I say that 0.75 is the value for the frequency of P, no, P means A. The frequency of A cannot be only 0.75. Why? Because A is also present over here. So you have to say that the frequency of allele, please be very focused now, the frequency of allele A is going to be capital A plus A. Capital A plus A. So let's find it out. The frequency of P or capital A is going to be 0.75. Now divide the 15 by 2 because there is one recessive allele. So divide the 15 by 2. 0.75 plus 0.075 because it would be divided, 15 should be divided first into, if I divided it by 15 divided by 100, it would be 0.15, and then I would be dividing it by 2, so it would become 0.075 because I have to only present the A, not the small a along with it. So the total value of P, not P square, remember this is the value of P square, this is the value of P square. I need to find out the value of P. So if I need to find out the value of P, I need to add both of them. 0.75 plus 0.075, that would be, sorry, I put it wrong, 0.75 plus 0.075, that would be 0.825. Now with the same method you are going to find out the probability of recessive allele, which means Q. Now the probability of recessive allele, probability of two recessive allele is going to be how you are going to do it. Q or recessive allele is going to be Q square plus Q. Now this is Q square. Now you have to divide it by 100 to mention them between 0 to 1. So dividing it by 100, 0.1 plus this allele is also going to be 0.075. So the answer is going to be 0.1 plus 0.075, that is going to be 0.175. Okay, now you have got it. P equals to 0.825, Q equals to 0.175. all right. Okay, now you are finding out actually the values that are expected. Remember, you are finding out the expected values. Now if I say, now you have got these observed value for P square plus Q square and 2PQ. 2PQ is recessive. So now I am going to represent the expected values. What would you do from here? You would perform P square, and P square means PP, which means 0.825 whole square, and 0.825 whole square means 825 whole square means 0.68. Let's find out Q square, which means QQ, and this is the expected. So 0.175 whole square, 0.175 whole square means 0.03. Great. Now what about the capital A small a? Remember, the capital A small a means one allele came from the sperm and the other allele came from the egg. Or small a capital A means this allele came from sperm and this allele came from egg. If I tell you both of the possibilities are from egg, sperm, and these both are the possibilities from the egg. So what does it mean? You can say that the capital A small a or small a capital A can be represented by PQ into QP, which would become 2PQ because of both the chances, which would become 2PQ. Now 2PQ equals to 0.175 into 0.825. It is going to be 0.144. 0.144. Now these are the values. You can even I did not multiply it with 2. I'm sorry, I did not multiply it with 2. 0.175 into 0.825 into 2, it is going to be 0.288. Now for your satisfaction you can add them. 0.68 plus 0.03 plus 0.288, it is going to be almost 1. So these are your expected values, expected allelic frequencies, or you can say expected genotypes. Now you see, this is how you are going to find out the expected genotypes. First step, let me tell you what is the first what the first step is going to be. The first step is going to be find out the values of individual alleles. Okay, you got that. You found it out, P and Q, values of P and Q. Then when you are done finding out the values of individual alleles, then you will multiply the expected genotypic values, expected genotypes, which means what you got expected P, you would turn it to P square and you would find a value X. Then expected Q square means you would multiply both Q values that you found in step 1, because you found the values of allele P and Q in the first step. Then you found the values of genotypes, and 2PQ means XY into 2. So in the end, when you are done finding out the values of expected genotypes 3, now you would multiply it with 100. Why? To put the data into table, because all of these values are between 0 to 100. So to put them into expected one, you have to multiply it with 100. If you multiply it with 100, it is going to be 68, it is going to be 3, and this one is going to be 28.8. So now let me write it. The value of AA, expected value of capital A is 68. The expected value of small a is 3, and the expected value for 2PQ is 28.8. Now let's do O minus E. Observed is 75 and expected is 68, so it is going to be 7. Then observed is 15 and expected is 28.8, it is going to be minus 13.8. And observed is 10 and expected is 3, so it is going to be 7. The square is 49, 13.8 whole square would be 190.4, and 7 O minus E, 7 whole square, it is going to be 49. Let me confirm the value, 13.8 whole square, 190.44. Alright, now we would be dividing it by expected values. 49 divided by 68, 0.72. 190.4 divided by 28.8, that is going to be 6.6, and 49 divided by 3, that is going to be 16.3. now our formula for chi-square is equal to chi-square sigma of O minus E whole square over E. So O minus E whole square is over here, so let's find out the x square, that is going to be 16.3 plus 6.6 plus 0.72, it is going to be 23.62. and what was your value? To test the hypothesis the population is at Hardey Weinberg, the scientists performed a chi-square survey. Which of the following value is closest to the chi-square? This is the value that is closer to the chi-square, 23.1. I hope you got it. Thank you very much.